And please, Hannah, show us your presentation.
Thank you, Matteo. Thank you for the invitation and for organizing this very interesting webinar. And thank you to Hepatoma Research for hosting us. So I don't seem to be able to. Okay, so these are my disclosures. I wouldn't go into the details of cholangiocarcinoma because we have heard already about it. What I really think is very important to highlight is that even though we classify cholangiocarcinoma based on its anatomical location, these are very heterogeneous types of cancer. They all develop from the biliary epithelium, but we know that the molecular, inflammatory, and the response rates and the therapies are very different. On the sake of time, I will focus on intrahepatic cholangiocarcinoma. And as you can see here very briefly, even in this type of cancer, those that develop from small bile ducts and those that develop from large bile ducts present a very different morphological aspect. The first studies highlighting the genomic heterogeneity of cholangiocarcinoma date back to the early 2010, in which Nakamura and other Japanese colleagues highlighted that the mutational burden of the tumor is highly associated to the prognosis of the patients. Nowadays, after a decade, NGS has given us a lot of information, and we know that we base our therapies on these mutational classes for cholangiocarcinoma. The important thing is that this mutational status of the tumor seem to correlate also to the inflammatory characteristics of the tumor and the immune response of intrahepatic cholangiocarcinoma. So although this information comes from bulk RNA sequencing, it gives us a lot of information because inflammatory tumors, although are a minority, are known to have a much better response. We did a couple of years ago a very simple study analyzing 50 intrahepatic cholangiocarcinomas that were resected, and with immunohistochemistry, you can see that the amount of T cells that are presented within the tumor strongly correlate with the prognosis. Colleagues from France were actually able to identify 4 types of cancer based on the immune microenvironment, again, analyzed by immunohistochemistry. I would highlight that some of them are very, very cold tumors. They call immune desert tumors. They seem to have almost no inflammation within the tumor. Some of them are very hot. Some have a more myeloid inflammatory pattern. We don't know exactly what drives this different type of inflammatory component, but we do know that it does correlate with the prognosis. Again, more recent study identified also the characteristics and the expression of immune checkpoints in intrahepatic cholangiocarcinoma. And again, the authors showed that some tumors have a very strong immune infiltrate. They are cold tumors, they are hot tumors, but again, we don't know why some present the one type of immune infiltrate compared to others. So we can conclude that intrahepatic cholangiocarcinoma is a highly heterogeneous cancer, even in its tumor microenvironment, that we know that there are different classes of intrahepatic cholangiocarcinoma, but we don't know exactly the characteristics of this inflammation, and most important, what is the interaction among the different cell types that are contained within the tumor. So we decided to perform a first study looking into the tumor microenvironment of intrahepatic cholangiocarcinoma, and we focused on T cells. The study is designed as follows. We have a first part of the study in which we isolated CD45 cells from the tumor, the peritumor, the perfusate of the non-tumor tissue, and the PBMCs. We did multiparametric FACS in order to identify the specific tumor populations that are contained in these tumors. After that, we did RNA single cell transcriptomic analysis using samples of cholangiocarcinoma from the tumor and the peritumor in order to identify the immune signatures that drive the differentiation within these tumors. And again, with the outcome of giving a clinical correlation with the outcome. I will try to be brief. As you can see here, we were able to identify different clusters of T cells, both CD8 and CD4. I would like you to focus on the fact that the perfusate and the peritumoral seems to have a similar composition, and that within the tumors you find tissue-resident T cells, both CD8 and CD4, but a high number of Tregs. Importantly, although the tissue-resident T cells are more abundant than the other components, these are still low compared to other types of tumors, suggesting, again, as already known by previous literature, that the cholangiocarcinoma is a prevalently cold tumor with very limited infiltrate. Since the Tregs were very abundant, we wanted to investigate them a little further and see what are the characteristics of these cells. As you can see here, they all express FOXP3, but we found a highly heterogeneous population with some of the T cells that were activated and some of them that were quiescent, suggesting that the this Treg population that we found within the tumor is actually heterogeneous. Moving into the transcriptomics analysis, in which we wanted to see which were the signatures that were activated in this type of cells, we showed that T cells and NKT cells were differentially present within the tumors compared to the peritumors. T cells are more abundant. The T cell class was actually divided into 10 different type of T populations, in which the Tregs, again, confirming what we saw on the FACS analysis, were much abundant within the tumor compared to the peritumor. Importantly, though, is not only that the T cells were more abundant, but within the tumor, the expression pattern of these cells was completely different compared to the peritumor, suggesting that the activation pattern of these Tregs in the tumor and the peritumor was completely different, and that there was some functional modulation of the T regulatory infiltrate within the intrahepatic cholangiocarcinoma. So this is a complex slide. I won't go into detail, but what the message is that if you look at the interactions that the Tregs have with the other types of T cells, you can see that there is an enhanced Treg-mediated immunosuppression within the intrahepatic cholangiocarcinoma microenvironment, suggesting that the Tregs drive an immunosuppressive environment within the intrahepatic cholangiocarcinoma. Again, this is another complex slide. I don't want to go into detail, but what I want to show here is that we identify through a complex biostatistical analysis that there are different transcription factors that are related to this hyperactivation of T regulatory cells within the tumor. Some of them were known, some other were actually poorly characterized in Tregs. One of these is MEOX1, which is a transcription factor that, as you can see here, was expressed almost exclusively in Tregs and is strongly associated to the expression of FOXP3. So based on this information and the fact that MEOX is actually regulated by a number of important transcription factors, we decided to focus on the activity of these transcription factors in regulating the pattern of T regulatory cells. What we did is we transfected Tregs isolated from healthy donors with the gene MEOX1 and with JFP marker, sorted the cells that express MEOX, and then analyzed their transcriptomic and epigenetic activation. This showed actually that genes that cells that have high expression of MEOX are able to have an important immunosuppressive pattern. Again, that not only they are immunosuppressive, but that there is a MEOX-dependent crosstalk between the Tregs and the microenvironment, which actually favors the immune responses against the tumor. And as you can see here, if you incubate conventional T cells with Tregs that have a high expression of MEOX, you obtain an important reduction of the infiltrate. This was correlated with the overall survival. So using an external data set of 150 intrahepatic cholangiocarcinoma patients, we were able to see that the signature is actually associated to worse prognosis. So what I can conclude for this talk is that intrahepatic cholangiocarcinoma is characterized by a poor immunogenicity with suppression of T cell responses, that the tumors are preferentially infiltrated by Tregs, showing a highly activated and immunosuppressive phenotype, that this phenotype is regulated by a number of transcription factors in which MEOX1 seems to have an important role that strongly correlates with the overall survival, and that could eventually use as a therapeutic target. A better understanding of the complex transcriptional architecture and the intracellular crosstalk of cholangiocarcinoma among different cell types will help to develop more effective therapies. I have chosen to present only a part of our work and the work of others for the sake of time. But as you can see, Anna, we have heard from other speakers the immune component of these tumors, and therefore also the knowledge of what the immunotherapy can bring to the patients is still far along to understand. It's very complex, and there is still a very large lack of information. I want to thank the people we have collaborated with for this work, Matteo and the group of the surgeons and the people in the lab, Cristiana, Michela, Barbara, and of course the collaboration with Enrico Lugli and his lab, the bioinformatics unit, and Diletta Dimitri. I thank you for your attention and happy to take any questions.
Thank you very much, Hannah, for the presentation. I think you did a very hard job for that paper because it was a very, very important paper. And in my opinion, we should go ahead and try to find a connection between immunotherapy and our findings, you know, in immunocontext of our patient. And probably that could be the way to better understand the response of this tumor if immunotherapy works or not. Thank you. Let's see if there is any question. Giuliano, thank you.
Yeah, thanks. Thank you very much. Very nice piece of work, and a lot of work too. I would like to take you, to a trip, hypothesis trip. Okay, take the tumor, and the tumor would produce chemokines, which recruit Tregs. Okay, and the Tregs would produce growth factors for the tumor. So your target would be the Tregs. So you would try to kill the Tregs and get the tumor die. So what do you think is the chemokine from the tumor cell, which could be, you could identify it recruits Tregs, and which are the growth factor produced by the Tregs, and you would find a receptor in the tumor cells?
Well, I don't think there is a white or black answer. I don't think there is one chemokine. As I showed, we have identified one transcription factor that is actually influenced by the expression of a lot of transcription factors. So as we said, for immunotherapy, this is a very complex network. I think that there are other cell types, and I would really like to look into the interaction with, for instance, tumor-associated fibroblasts, which I think in cholangiocarcinoma are of huge importance. So I would like to have an answer for your question, because that would be crucial for our patients, but unfortunately I don't. I think there is a lot to do. What is important for me from this study is that we know very, very little about the immune infiltrate of these tumors. And we are using immunotherapy, and we are seeing some benefit in some patients, but we don't know what this response is based on. So unless we are able to decipher a little better what the immune network is about, we are going to be wasting the tools and the money, because these tools are very expensive, and the time of these people, just because there is no other option than trying.
May I just repeat? I want you to believe on Tregs.
I do believe in them.
If you believe on Tregs, then you just can imagine, just imagine, you should have John Lennon in the background. Just imagine, imagine what is the chemokine, what could be the chemokine recruiting Tregs. Okay, and if you check the database and look at what the Tregs produce, what do they produce, which could, resemble a growth factor. It would be just, in my opinion, worth to check this hypothesis. That's
no, absolutely. We are looking into it. Absolutely. Thank you for this.
Very nice work. A lot of work. Thank you.
Thank you, Giuliano. Mr. Azzam?
Hi, Anna, well done. Really, I am proud of this, what you are doing. Did you believe on microbiome or not?
I do, absolutely.
Okay, why not? Very good, very good. Thank you very much. Thank you very much. Why not to add?
No, absolutely. I think there is a lot to be explored in cholangio, but I think the bile microbiome is going to be giving us a lot of surprises.
Okay, thank you very much. This is the one. This is a message I want to finish it today. Thank you very much. Well done. Well done.
Sure.